Good. So I think we'll go ahead and get started here. So it's my great pleasure to introduce Matt Taliaferro from the University of Colorado Anschutz Medical Campus. And so like many great scientists, Matt trained in the field of RNA splicing. in this case, alternative splicing with Don Rio as a graduate student. And there he did quite a bit of work on some very core processes in alternative splicing and core factors, including things that bind the 3' end of introns using Drosophila as a model system. And I would say like many people training or great scientists, he also trained in a model organism. He then went on to Chris Burge's lab before starting his own group at University of Colorado Medical School in 2017, I believe, where he's now an assistant professor. And so he's done a lot of really nice work in his own lab. I think it was about a little over a year ago when we were thinking about who to invite, and he had this great paper come up on bioRxiv that caught my attention on a great synthesis of a chemical biology tool called the Halo tag with RNA sequencing called HaloSeq, and that caught my eye, and I was really glad that we were able to invite him here to talk to us about his current research in RNA localization, RNA sequencing, and visualization of RNAs in cells. And I'll just end up, I think he must also have a great sense of humor, because he's also developed some software that he calls LabRat for looking at polyadenylated RNAs. And so for all the lab rats, you can now use LabRats to study your RNAs. So with that, let's thank Matt.
Thank you, Aaron and Melissa, for the invitation. It's so nice to see people in person, and I've had just a great day meeting with many of you and talking about all of the really cool stuff that's going on here. So as Aaron said, our lab studies RNA localization, and so what this is the spatial regulation of the expression of genetic information. And so if you consider a cell like a neuron, and I've kind of, everyone knows generally the morphology of a neuron. It's kind of drawn here. It's got these long, thin processes. And let's say this cell wants to make a protein that's got to do its thing at the end of one of these processes. And there's kind of two major ways that this protein can get out here. It could be made in the cell body, and then once this protein is made, it could be shipped, out to the tip of this process. And this totally happens. This is a totally valid way to do this. But alternatively, what you could do is, instead of shipping the finished protein product, you could ship the RNA out to this location and then translate the protein on site. And so as soon as this protein is made, it's immediately at the correct place. And so this is a really dynamic process, and it's one of the reasons that I was really drawn to it. And so what you can see here are individual RNA molecules that, in this case, encode beta-actin. You can see that they're kind of zipping up and down these neuronal processes. And so it's this incredibly dynamic process. It's really important in a lot of different settings and in a lot of different organisms. So it's required for mating type switching in yeast. One of these RNAs has to be No, I don't care about that new version. Okay. So one of these RNAs has to be localized to the bud in order for this to happen. It contributes, as many of you know, to quite a bit of developmental patterning in the Drosophila embryo. So each one of these embryos here is stained with a fish probe for a different RNA. And I hope that you can appreciate that all these different RNAs have wildly different spatial localizations within these embryos. And mutations in many of the RNA binding proteins that regulate this process are associated with neurological disease in human. So despite this, extreme utmost importance, the sequence determinants of how these RNAs get to where they're going are really poorly understood. They're really only known for a handful of transcripts. And part of that is because this localization information can be stored in either the primary sequence of the RNA or within its secondary structure. And if it's within the secondary structure, that makes it much, much harder to find. Okay, so for the RNAs that we do know how they're getting to where they're going, this is how it works. So we've got a beta-actin mRNA here, and what happens is that there's a particular sequence within this 3' UTR that's called a zip code. So some of the younger students in the audience may not remember, but back in the day we used to write these things called letters, and letters would go in envelopes, and in the envelope you would put 5 numbers that would tell the mail carrier, where to send that envelope, right? And so those things are called zip codes, and so very analogously here, these sequences that are telling the RNA where to go are called zip codes, all right? So there's a zip code within this 3' UTR of this RNA, and it's bound by this protein called ZBP1 for zip code binding protein 1. And when that happens, this thing recruits motor proteins and other cytoskeletal elements, and when all that happens, then this RNA is correctly localized. And one of the ways that we know this is because if you mutate or delete or otherwise kind of mess with the zip code, you don't get ZBP1 binding, you don't get the recruitment of all that other stuff, and this RNA is not where it's supposed to be. So although there are thousands of these transcripts that are asymmetrically localized in a variety of cell types, we know very little of this regulatory language regulating this localization. And by regulatory language, I mean the identity of these cis element zip codes and the transfactor RBPs that bind them. And so that's really the core of what our lab is trying to figure out. What are these zip codes and what are the RBPs that mediate their transport? So classically, how this has been figured out has been with taking single localized RNAs and then asking, okay, how did it get there? You know, what sequences within this RNA are responsible for transport? And so what people have often done is take pieces of these RNAs and put them onto a reporter construct, and now ask, is that reporter construct at the same location where this endogenous RNA was originally? And if not, well, then that tested sequence must not be sufficient for transport. But if yes, then what you can do is test progressively smaller sequences, you know, each time trying to really narrow in on what's the minimal sequence required for the transport of this RNA. And so this is great work, you know, this is really the foundation of the field, but it's extremely laborious. I mean, because you got to go through multiple iterations of this testing, you know, fusing and all that kind of stuff. And so really, figuring out how this works for one of these RNAs is kind of like probably the majority of a thesis project, right? It's just extremely laborious. And that's probably one of the reasons why we don't know for most RNAs how it works. It's just because it's too much work. Furthermore, after you've gone through all this work, what you've figured out is what's responsible for the localization of this one RNA, right? Which is great, but it's really hard to generalize that, what you found there, to other, you know, to other localized RNAs to try to figure out how they're getting to where they're supposed to be. So we wanted to come at this problem from kind of the other direction, and that's to take more of a genomic type approach. And so our idea is to isolate all of the RNA from a given subcellular location. In this case, the subcellular location is the right side of this oval, and then sequence it. And by looking at those sequences of those localized RNAs, ask, do they have common features or common you know, structures that might be behind why they got to that location in the first place? And so not only that, we can cross this by taking RNA binding proteins that we think might regulate this, knocking them out, and now asking what's mislocalized. And so that experiment tells us two things. In this case, these orange RNAs are mislocalized if you don't have the purple protein, right? So this tells us that the orange RNAs depend on the purple protein for transport. And if you look at the sequences of the mislocalized RNAs, you'll often find that they're enriched for certain things, which are most likely the binding sites of the RBP that we knocked out to begin with, right? So this tells us a lot about the mechanism of how these things are getting there. So if we want to do this, we need a way to isolate subcellular transcriptomes and sequence them, right? So how can we do this? So we started with neuronal systems, and I'm not a neuroscientist, so these are not primary neurons, although all the experiments I'm going to tell you, we have recapitulated in primary neurons, and all of our main findings hold. But because I'm just your garden variety molecular biologist, these are, you know, cell lines that we can grow in a dish in the lab easily. And so I hope you can see that in culture they make these long, thin processes, okay? So these are a couple mouse cell lines. And so what we can do is we can grow them on these porous membranes. And the pores in these membranes are big enough such that the projections or neurites of these cells grow down through to the bottom, but they're small enough such that the cell bodies stay on the top. And so after growing these cells on this membrane, what we can do is just scrape the top of the membrane, and that separates the cell bodies from the neurites. We can isolate RNA from both of those fractions and sequence it. And then we know, which RNAs are in the cell body fraction, which RNAs are in the neurite fraction. And this is just a Western blot to show you that this fractionation really works. Histone H3 should, of course, be only in the cell body and not in the neurite, and that's where we see it. Okay, so what does this data look like? So for each gene, we can define something that we call localization ratio, which is just its abundance in the neurite fraction compared to its abundance in the soma fraction. And here I'm showing you a plot where each dot is a gene, and we're comparing this data from our two cell lines, CAD cells and N2A cells. And I hope you can see that there's a really strong correlation between these two cell types. And so what we think, what we're getting is very reproducible even across cell types. If we just ask, what is this neurite-enriched RNA, we can take some of the examples in the literature that people have identified previously as neurite-enriched RNA. They're neurite-enriched in our samples. And just generally, they tend to encode for ribosomal proteins and mitochondrial proteins, both of which, those things are consistent with the literature about the subject. Okay, so now we've got our system set up. We can analyze subcellular transcriptomes. Now we're going to knock out an RNA binding protein and ask what transcripts are mislocalized. And so we need to pick an RNA binding protein to do this. So the first one that we picked was FMRP, which is encoded by the gene FMR1. So loss of FMRP is associated with Fragile X syndrome in humans, which is associated with intellectual disabilities, including other things. Knockout mice show similar phenotypes. And this protein is known to move along the cytoskeleton and transport a few RNAs, but really which RNAs depend on it are kind of unknown, and how FMRP is recognizing those localization targets is also unknown. And by the way, it also is a translational repressor. So these are the questions that we kind of set out to ask. Which transcripts depend on FMRP for transport? How does FMRP recognize those transportation targets? And are there any molecular relationships between the loss of FMRP and Fragile X phenotypes that we see in patients? And so this was the work of our first graduate student in our lab, Rayanne. This is all published, so I'm only going to spend one slide summarizing the main results. She made an FMRP knockout line using CRISPR, and then fractionated and sequenced it, and what we found was this. There were a whole bunch of RNAs that in the wild type cells are in the neurites, but once you get rid of FMRP, they are no longer trafficked to the neurites. And if you look at those RNAs, they are super enriched for having a G quadruplex structure in their 3' UTRs. So from this, we infer that FMRP is recognizing G quadruplexes within messages, and when it does that, it transports them to the neurite. So not only that, using some molecular genetics, she found the domain of FMRP that was responsible for this. We took iPS cells from Fragile X patients, differentiated them into neurons, and did the same fractionation and sequencing. The same messages that are mislocalized in our mouse culture system are also mislocalized in human Fragile X patient neurons. And she was also able to identify some separation of function mutations that separate the translational regulation role of FMRP from its localization regulatory role. So, this was 4 years of work summarized in a single slide, but the reason that I tell this to you is because we can do this for any given RBP. And in fact, we're working with others now. So we kind of got this recipe going where, we knock it out, what's mislocalized, what kind of motifs are enriched in mislocalized RNA, that kind of stuff. Okay, so this is all great, it works really well, but anything that we find here is necessarily biased because we can only find RNA localization mechanisms, in this case, that act through FMRP, right? Because that's the protein that we knocked out. So is there a way to instead directly, and in an unbiased manner, identify RNA sequences that have the ability to act as zip codes, basically, okay? And so this is the work of a postdoc in our lab, Ankita. And so if we go back to this model that I showed earlier, generally these zip code sequences that are known so far tend to be found in 3' UTRs. So if we're going to go out and try to find new ones, let's start looking in 3' UTRs, right? So we've done this kind of neuron fractionation and sequencing experiment 30 times, I think. And so we know which RNAs are very reproducibly enriched in these neurites across a variety of samples. And so that's just kind of shown here. So we took 9 genes that we saw very reproducibly enriched in neurites, and 4 control genes that were not enriched in neurites. And we took their 3' UTRs, and we asked, or basically we're asking what sequences within these genes are behind their localization to neurites. Perhaps unsurprisingly, in their 3' UTRs, the 3' UTRs of these localized genes are more conserved than you might expect. So this is how we are kind of assaying RNA localization. It's a qPCR-based assay, and we're going to use it a lot, so I'm going to spend just a little bit of time walking through it. So we can site-specifically integrate with Cre recombinase a construct that looks like this, where we've got a bidirectional promoter, one side of which is driving firefly luciferase, the other side of which is driving renilla luciferase. We attach whatever UTR we want to test the RNA localization activity of to the firefly luciferase RNA. And then we do our fractionation, like I talked about earlier, isolate 2 RNA samples, one from the cell body, one from the neurite. And in each fraction, in each RNA fraction, we can quantify the firefly to renilla RNA ratio using qPCR. And so if we compare that ratio of firefly to renilla in the cell body RNA to the ratio in the neurite RNA, the ratio of these 2 ratios tells us how localized that transcript is. Okay, so we took the 3' UTRs from the genes I talked about a couple slides ago, attached them onto firefly, and asked, did that make that firefly transcript go to the neurite? And for 6 of the 9 genes that we tested, it did. Okay, so for all of these here, and importantly for none of the controls for the RNAs that were, the endogenous RNAs are localized, did the 3' UTR have activity. So we know somewhere within these 3' UTRs is a zip code sequence. Is a sequence that has the ability to take this RNA to neurites. So that's great, but we want a little bit better resolution than somewhere within the whole 3' UTR. So to get at this, we designed a massively parallel reporter assay where we took these 3' UTRs and we made a whole bunch of oligos, each of which was 260 nucleotides long, and we designed them such that they tile across these UTRs. All right, so we made it so there's 1 oligo every 4 nucleotides. So this is quite tight tiling. It ends up being about 8,100 oligos in total across all of the UTRs that we're testing. So what we do then is take these oligos and integrate them into the same reporter transcript that we were talking about earlier. And so now we have 8,100 different reporters, each of which is exactly identical except for the sequence of the oligo that we put in there. Okay? Now we take that library of oligos, express it in cells, do our same fractionation and sequencing, and then for each oligo, count how many times we see it in neurite RNA, count how many times we see it in soma RNA, and those that are more abundant in the neurite than the soma must have the ability to take this reporter to the neurites. So for rigor and reproducibility, we incorporated these oligos into 2 different reporter scaffolds, one of which encoded firefly luciferase, the other which encoded GFP, and we integrated them and did this experiment in our 2 different neuronal cell lines, again, CAD and Into-A. This is just to show you that after we integrate these reporters site-specifically into the genome of these cell lines, we can still detect 96% of them. So we're kind of assaying pretty much every oligo that we set out to assay. So this is what the data looks like. So if you compare, so we did 4 replicates, and so if you compare the oligo abundances in neurite replicates to each other, they look more like each other than they do to the soma replicates. So there is some kind of reproducibility or reproducible activity that we are detecting here. And then we wanted to ask, are the results consistent across the 2 cell lines that we tested? So our CAD cell line and the Into-A cell line. And so what we did is we took the oligos that we observed were enriched in the neurite RNA data and asked, where are they in the same data but from the Into-A cell line? You can see they're way more neurite enriched than we would expect. And we did the same thing comparing our results across our 2 reporter scaffolds, the firefly luciferase scaffold and the GFP scaffold. And we see the same thing. So if an oligo is able to drive localization of one reporter RNA, it's able to drive the localization of the other reporter RNA, and it's able to do it in 2 independent cell lines. So we think these are pretty robust general targeting RNAs. This is what the data generally looks like. So on the x-axis is the position that an oligo was drawn from, so across the UTR. And on the y-axis is the enrichment of that oligo in the neurite. And so what I hope you can see is that along some of these RNAs, there's a clear peak of activity, and that peak occurs in both cell lines, so in both colors here. And so basically what this is telling us is that the sequences that are able to drive these RNAs to the neurites lie under these peaks. And so for many of these UTRs, we were able to pretty clearly define peaks here. This is a control RNA where the whole 3' UTR didn't have activity, and we can see that there's no peak anywhere within this 3' UTR. So now we're able to say, it's not the whole UTR that has activity, it's this sequence right here that has activity. Right, I'm going to skip that. Okay, so well, actually I can't skip that, I'm sorry. Okay, so remember we have one oligo every 4 nucleotides. And so we defined something that we call an active window, which is just if this is our kind of string of active oligos, this active window is just the union of all the sequences that had activity. And so we can ask now, what are kind of the sequence properties of these active windows? And the first thing that really jumped out to us was that they were super enriched in A's and G's. And we couldn't derive any more information-rich, complicated k-mer than that. We can just say they have way more adenosine and guanosine residues than we would expect. And that's kind of shown here. So the rest of the UTR, has some level of A and G richness. But if you look at the active windows within those UTRs, they have way more A's and G's. And again, plotted across the UTR, here I'm plotting the fraction of A and G sequences at a given location. And the sequences that had the ability to drive localization, you can see lie under just really rich AG peaks. We then asked, are these active windows more conserved? And the answer, surprisingly, was no. They're actually much less conserved than we would have expected. However, if we look in the human orthologs of these UTRs, the human orthologs also have a very AG-rich region. And so when you think about UTR regulation, a given regulatory element doesn't have to be at the same orthologous position within the UTR in order to be active. So it may not be conserved if you look at something like FastCons, it just has to be present within an orthologous UTR. And that's exactly what we see here. However, just having a high AG-rich window alone is not sufficient to drive RNA localization. So here, if we look and just bin genes into those that either have an AG-rich window in the UTR and those that don't, there's no difference in their neurite enrichment. So there's something beyond simple AG richness that's driving this, but we don't understand that yet. And that's one thing that we're working on now. Okay, so we took kind of the most enriched oligo within those peaks of activity that I showed before, and we just asked by single molecule FISH, is this able to drive the reporter to the neurite? And it was. And so if we just have the reporter on its own, with nothing fused, we don't see much of this yellow reporter RNA in the neurite. But once we add the peak oligos that we derived from these two different UTRs, I hope you can see that there's a lot more of this RNA within the neurite. So these oligos are sufficient to take this transcript to drive it out to these neurites. Very interestingly, we then compared the activity of this peak oligo to the activity of the full 3' UTR that it was derived from. And these peak oligos often had very different activity than the full 3' UTR. So implying that yes, these are sufficient, but there are other sequences within the 3' UTR that are required for full activity, even though those other sequences aren't sufficient on their own, right? Because we tested pretty much every possible 260-mer that you could make within that 3' UTR. To ask if these oligos were necessary, we made the whole 3' UTR, but just took out that 260. And if you take out that 260 nucleotides, it doesn't work anymore. So these oligos are both necessary and sufficient for the localization of these RNAs. Right. Okay, so then we wanted to ask, can we get a little bit better resolution than just this union active window? And so we reasoned that maybe what's really driving this is not the union between all of these sequences, but the intersection. So what's shared among each oligo that's localized. And so we called that the minimal element, because we went in thinking that it would be the minimal element. So now it's not a minimal element, because it actually doesn't work. So, what this means is that we've got an oligo that's 260 nucleotides long that is active. we can take a subsequence of that, this minimal element, that is, depending on the element, between 50 and 150 nucleotides long. That doesn't work. So the minimal size of these RNA elements is somewhere between 150 and 260. So that's big, like for RNA localization, like not big as in important, I mean big as in large, right? So it's either true that the actual minimal element is quite large, or that somehow the context surrounding this element matters. And this is, again, something that we're following up on now, a current student is looking at. And so, kind of like I said in the beginning, the size of these elements is consistent with secondary structure really being involved, and not just primary sequence. All right, what is the RNA binding protein that's recognizing these zip codes to mediate their transport? So to try to get at this, we transcribed these RNA elements, put a biotin on them, put them into cellular extract, pull it back out with streptavidin, and with mass spec ask what proteins are bound to these oligos. Very reproducibly, one that was bound was a protein called unkempt. It comes from the fly gene unkempt. And in fact, we could detect that also by Western blotting. Again, if we put our active oligo element into extract and pull it back out, we find that unkempt is associated with it. So does this association actually matter? So to test this, we again assayed the RNA localization of our constructs, but either with or without knocking down unkempt. And it turns out if you knock down unkempt, these oligos don't work so well anymore. So unkempt is required for their efficient localization. So we think here's a case where we've both identified the zip code cis element as well as the RNA binding protein that mediates them. So this is kind of one check off the list. We've identified a pair. So to summarize this part, we used an MPRA to find sequences that are both necessary and sufficient for RNA transport. These sequences are very AG-rich and really quite large. And the RNA binding unkempt is required for their efficient activity. So everything that I've told you so far is in neuronal systems. But there's this whole big, wide RNA localization world out there that is not in neurons. But we can't really get at them because our system, relies on the morphology that the neurons have, right? This transwell porous system. So, there's tons of RNAs that are localized in systems that are not amenable to this type of fractionation. And so this kind of led us to a question that is, I think, partly profound and partly very silly, which is kind of shown here. So if you think about an RNA process like RNA stability, let's say that in a neuronal cell type, I've got an RBP here that recognizes some element within this 3' UTR. And when that happens, that causes the RNA to be degraded. If I go to a completely different cell type, like an epithelial cell, and I've got the same RBP expressed and the same RNA expressed, generally what's going to happen is this RBP is going to, again, bind the same RNA and lead to its degradation. Okay? But RNA localization is, potentially a little different. So let's say in neurons, I've got an RBP that recognizes some element within a 3' UTR. And when that happens, it causes this RNA to be transported to neurites. And so now back to the epithelial system, if I've got the same RBP and the same RNA, what's going to happen? Right? This epithelial cell doesn't have neurites. It doesn't have axons or dendrites. And so is it going to be transported? And if so, to where? So another way to say this is what's the spatial equivalent of a neurite in a cell that doesn't have a neurite, like an epithelial cell? Again, partly profound, partly silly. So to do this, we have to have a way to assay RNA localization in this epithelial cell, which again, this will not work. So we had to come up with a way to do that. And so we developed this RNA proximity labeling system that we call HaloSeq. And just very briefly, the way it works is that we express a halo-tagged protein that is localized to a very specific place within the cell. And so here it's fused to a histone. We then add a halo ligand that will attach itself to this halo-tagged protein. And this ligand is special because when you shine green light on it, it makes oxygen radicals that will label any RNA that's nearby, so within about 100 nanometers. This labeling is set up such that those RNAs will become alkynylated. We can then isolate all the RNA within the cell. The localized or haloproximal RNA will be alkylated. And we can then biotinylate this alkylated RNA in vitro using a click reaction, which allows us to then pull it out and sequence it. And so now again, we've got two RNA populations, one that was localized and therefore biotinylated, and kind of the total RNA population to compare it to. So this is just to kind of show you that this works. If we add our halo ligand, we can biotinylate RNA in vitro, but if we don't add our halo ligand, we can't. We tested this by fusing a halo domain to 3 different locations, to chromatin, to the nucleolus, and just broadly into the cytoplasm. The reason that we did this is because we know which RNAs are at each of these locations, so they're kind of like positive controls. Does this method work? All of this is to say that this method works. So for example, 7SL, it's known to be very localized to the nucleolus. We only really pull it down when we've got our halo protein fused to fibrillar, which is also nucleolar. And the rest of these are other positive controls that make a bunch of sense. So, okay, so this method works. Can we apply it to a system that's a little more interesting, like epithelial cells? And so epithelial cells, like neurons, are very polarized. They have an apical to basal axis. So the apical side often points to lumen, while the basal side points more to like blood vessels in the interior of your body. They create a tight layer. The intestinal cells that we used are important for digestion and nutrient uptake. And they're also important for immunity. And so, like I said, they have this very this very polarized axis from the apical side to the basal side. And there are proteins that reflect this apical basal polarity. And so, for example, the ezrin protein is very localized to the apical side of the cell. And this sodium-potassium ATPase is very localized to the basal side of the cell. So we're going to be looking at a lot of pictures that look like this. So these are just cells that we take a picture of from down below and then computationally kind of rotate them such that we're looking at them end on. So the apical side is always going to be on the top here, and the basal side is always going to be on the bottom. It's also known that there are RNAs that are asymmetrically distributed along this axis. But again, what we don't know is how these RNAs are getting to these locations. So it's back to the same exact question, what are the cis elements and trans factors that are causing these RNAs to be localized across this axis? This is the work of, again, this very talented graduate student, Rayanne. And so she's set to work applying our halo-seq technique to these epithelial cells. So she made a halo fusion that was specifically localized to the apical side of the cell, and she made a halo fusion that was specifically localized to the basal side of the cell. And she defined this metric that she called apical bias, which was in the halo-seq purified RNA, the abundance for a gene in the apically labeled RNA divided by its abundance in the basally enriched RNA. And so after sequencing these RNA populations, she saw that there were many RNAs that were enriched either apically or basally. And she took 3 of these, shown here, to kind of follow up on. So 2 of these, this collagen protein and OSBPL3, were enriched in the apical halo-seq experiment, while the other was enriched in the basal halo-seq experiment. And she then did single molecule FISH for each one of these RNAs, and sure enough, by single molecule FISH, the RNAs that were apical by the halo-seq experiment were also, visually apical, and the same thing for the basal RNAs. Okay, so one thing that immediately kind of struck Ray is that if she looked at all of the genes that encode ribosomal proteins, so these are not our RNA, they're mRNAs that encode protein components of the ribosome, she saw that as a class, they were much more basal than she would expect. Much more basal than all RNAs. And so we got to thinking, what is regulating the transport of these ribosomal protein RNAs to the basal pole? Maybe it's something that they all have in common. Maybe it's an element that they all share, since they are, as a class, localized to one location. And so one thing that they do all share is something called a top motif. And so a top motif is just a run of pyrimidines that are almost always found at the 5' end of a message. And so, maybe this is what's behind the localization of these RNAs. So to test this, what she did was take the top motifs from 2 of these RNAs and fuse it onto our reporter transcripts that we had used in the neuronal system. So 2 RNAs, RPL7 and RPS28, and as control, she also made mutant top motifs where every pyrimidine was just mutated to a purine. She expresses these in cells and then asked by single molecule FISH, where is the reporter RNA? So if we don't express our reporter, there's no reporter RNA signal, which is good. If we just express our reporter fused to nothing, so to no top motif, I hope you can see it kind of fills the cell from apical to basal. But if she puts the 5' top motif from RPL7 on there, you can see that the RNA is now much more towards the bottom of the cell. And this is actually quantified here. Again, so the top motif makes this RNA be more basal. But for the mutant, that's not true. And the same thing was true for the other top motif from the other gene that she tested. So these 5' top motifs are sufficient to drive an RNA to the basal end of these epithelial cells. So now we can go back to our kind of silly question here. So we've kind of done it in reverse. We've identified an element that when it's expressed in epithelial cells, it causes an RNA to go to the basal end of these cells. What happens if we take that same RNA and we put it in a neuronal system? So now we're asking what's the spatial equivalent of the epithelial basal compartment in something that doesn't have an epithelial basal compartment, like a neuron. Right? So we take that same reporter and we put it in neuronal cells. Where does it go? So this is the same experiment. So now just our blank firefly RNA on its own doesn't really get out to the neurite. But once you put the 5' top motif from RPL7, there's a lot of it in the neurite, but not if you add the mutant top motif. And the same thing is true for RPS28. And again, this is quantified here. Wild type top motif, very good at taking this RNA out to the neurite. Mutant top motif, not so much. Okay, so the same reporter is both basally localized in epithelial cells and neurite localized in neurons. So perhaps these are kind of like spatial equivalents, right? All right, so what RNA binding protein is behind the localization of these top motifs? So it was known from other literature that top motifs are often bound by a family of protein called LARPs, for law-related proteins. And in mammals, there's like 7 or 8 of them, I think. So we did a targeted little siRNA screen against all of those LARPs and asked which of them are required for the localization of this reporter out to these neurites. And it turned out that one of the LARPs was required for this, is this protein called LARP1. So if we knock down LARP1, a reporter that used to be very neurite enriched is no longer neurite enriched. Okay, so efficient 5' top transport to these neurites requires the protein LARP1. Interestingly, in the cells that we knocked down LARP1, their neurites were much shorter. So maybe there's something phenotypically or morphologically that LARP1 is doing that helps these neurites form. So this is quantified here. You knock down LARP1, the neurites are about half as long as they were before, and cells just don't really make as many neurites. So given that these 5' top motifs are found in almost every ribosomal protein RNA, perhaps their loss from the neurite, due to the loss of LARP1, inhibits neurite formation. This is very speculative. I mean, we can't draw this conclusion directly from this data, but this is to say that maybe RNA localization actually matters, because there's not a whole lot of defined cases where that's true. So anything that smells like that, I'm going to highlight it. All right. Neuronal cells, 5' tops require LARP1 for an RNA to get to neurites. What about the epithelial cells? Is LARP1 required for the localization of top motif RNAs to the basal pole? We can't do siRNA in our epithelial model, but we can do CRISPR. And so we just went ahead and we knocked out LARP1. And so we knocked out LARP1, and then we either rescued it with a DOX-inducible rescuing copy of LARP1 or with GFP, so it's still a knockout. All right, so if we now look at our reporter where we rescued the knockout with LARP1, the reporter is still very basal. If we rescue the knockout with GFP, it's now not so basal anymore. And the same thing is true with RPS28. Again, quantified here. If there's LARP1 in the cell, these reporters are very basal. If there's no LARP1, they're not basal. So again, these 5' top motifs require the presence of LARP1 for their localization. In these two very different cell types, neurons, epithelial cells, something I might have forgotten to mention, our neuronal system is mouse, these epithelial cells are human. So all of this stuff is cross-species too. Okay, so something that was sort of curious to us is that in all of these ribosomal protein mRNAs, the top motif is always at the extreme 5' end, always. And so, like, does that matter, right? Does it have to be there? And so to test this, we took the same top motif and we moved it from the 5' UTR to the 3' UTR. And now asked, is our reporter localized? And the answer is no. So you put the top motif in the 5' end of the transcript, it's basal. Same motif in the 3' UTR, not basal. Same thing is true for neuronal cells. 5' top motif, it's neurite enriched. 3' top motif, not. So that's, you know, kind of weird, right? So it turns out that it's known that for LARP1, or that LARP1 has at least 2 different RNA binding domains, one of which recognizes top motifs, but the other directly interacts with the 5' cap. And you actually need both of these domains engaged at the same time with RNA in order to get efficient binding. So if you take this top motif and put it in the 3' UTR, LARP1 can't bind it anymore, and we think that's why it's not able to be localized. All right. So are these 5' tops the only examples of these kind of dual targeting neurite basal RNA elements? You know, and of course, if I'm asking this question like this, then the answer is no. So if you remember from the first part of the talk, where we did this MPRA, we identified RNA sequences within some 3' UTRs that had the ability to take RNAs out to neurites. So we can take these same elements, throw them in epithelial cells, and ask what happens. Right? So we made 4 constructs, just our blank reporter, one where we put the entire 3' UTR from net1, one where we just put the localization element that we identified from the MPRA to ask if this element is sufficient, and then one where we took the 3' UTR without this element to ask if the element's necessary. And so this is the kind of more or less the same neuronal data. The whole 3' UTR is very good at getting this RNA out there. The localization element on its own, pretty good, but the localization element is absolutely required. Now here's the data from the epithelial cells. The whole 3' UTR is very good at making something basal. The localization element on its own, pretty good. The whole 3' UTR without this localization element doesn't work at all. So these are very much mirror each other. And so this is another case where we've identified an element that is capable of both taking RNAs out to neurites and to the basal pole of epithelial cells. And so we can ask this question much more generally, much more broadly, transcriptome-wide actually, because for every gene from our neuron fractionation experiment, we know how enriched its RNAs are in neurites compared to cell bodies. And from our HaloSeq experiment, we know how enriched that same gene is, you know, either in the basal compartment or the apical compartment. And it turns out there's a pretty darn strong correlation in the negative direction here for the localization of these RNAs in the 2 systems. And the negative direction, again, meaning that an RNA that is neurite enriched in the neurons is basally enriched in the epithelial cells. This is a pretty darn strong correlation when you consider that these are 2 very different experimental setups. So this is the mechanical transwell fractionation, it's proximity labeling, and these are 2 different species, again, you know, human and mouse. So I think we can say with some confidence that the spatial equivalent of a neurite in an epithelial cell is the basal compartment. These 2 these 2 locations contain the same RNAs, or are enriched for the same RNAs. Okay, so how is, like, what's behind this, right? It's like kind of bizarre. So it turns out that in both of these cell types, the microtubule organization of these, like within these cells, is kind of similar. So within neurons, the plus ends of microtubules point out to the tips of these neurites. And in epithelial cells, the plus ends of these microtubules point down to the basal end, or to the basal compartment of these cells. So is microtubule trafficking behind what we're seeing in terms of like similar RNA trafficking? So if this is true, what we should be able to do is inhibit kinesin, and now our RNAs that were neurite enriched or basal enriched should not be that way anymore. So that's what we did. We took an RNA that was very basally enriched, and if we inhibit dynein, it actually becomes more basally enriched. But if we inhibit kinesin, now you can see it's kind of apical basal filling the cell. So this is quantified here. And then the same thing for the neuronal system. If we inhibit dynein, this reporter RNA still makes it out to the neurites. But if we inhibit kinesin, this RNA is not in the neurites anymore. And again, quantified here. So again, we've kind of defined these two spatial equivalents in cells that have very different morphologies. And what's behind this is the fact that they have similar underlying cytoskeletal architectures. So to summarize this last part, we've used HaloSeq in intestinal epithelial cells to identify RNAs that are asymmetrically distributed along this axis. 5' top motifs are sufficient to drive RNAs along this axis, specifically to the basal pole. LARP1 is required for the transport of these 5' top motifs in both of these cell types. And that neurites and the basal pole of these epithelial cells basically have the same RNA contents, implying that the similar mechanisms are populating their transcriptomes. So with that, I'd just like to thank the people in the lab, specifically Ankita and our first graduate student, Ray, who is very close to graduating and I will miss very much. Our collaborators here, you guys for inviting me. This has, again, just been like the best day in a long time. And all of you for showing up and listening. I'm happy to take any questions.
Thank you. I think we already have a lot of questions. We're going to start with Dave and work our way back.
Nice talk.
Thank you.
So why would a neuron want to localize ribosomal protein coding mRNAs to the neurites when proteins have to go back to the nucleolus, presumably to be incorporated into the ribosomes?
This is a great question. So we don't really know, but there is some evidence that ribosomes can be repaired kind of on site in, I think it was axons specifically. So, maybe if it's a sort of surface exposed or external ribosome protein and it breaks for whatever reason, it could potentially be replaced by a ribosome protein that was synthesized, on site without having to wait for it to come.
Although you did say it was most or almost all of the ribosomal protein messenger RNAs.
That's true. That's true. Which, if it's a protein within the middle of the ribosome, I don't know how you get to it, right? But this is kind of the feeling in the field so far. But that's it's still kind of a paradox.
Hi, beautiful talk.
Thanks.
So my question is, neurons have both dendrites and axons. And in your correlation, did you try to see whether some of your basal apical epithelium is actually differentially correlate with axon and dendrite?
Yeah, so with our, so like I said, I'm not a neuroscientist, and so I kind of, struggle with these more fine-grained real neuron type questions. Our system, the transwall system is not differentiating between axons and dendrites. And further, I'll just say that. So we can't, with our RNA-seq data, differentiate between those two. We do think that the cell lines that we use, their projections are more axon-like than dendrite-like because of some markers that they express. So we would think that our data is more kind of in line with the axon RNA content in a primary neuron than the dendrite, but we can't. So that's why I'm just using the term neurite over and over to kind of cover myself there a little bit. That's a great question, and the answer is we can't say.
Great. So we have a question down here.
Does this work? Maybe. Great talk. So I had two questions. One was any biasing on ribosomal RNA, rRNA, along with a message for the proteins.
So we don't normally look for that. I mean, it's definitely not in the sequencing data because we're actively trying to get rid of it when we're making, the RNA-seq libraries. I did a little bit of stuff as a postdoc looking at that. There was some crazy stuff, like I could see 45S unprocessed rRNA in the neurite. And getting back to your question, there's no reason that should be out there unless there's active, like, de novo assembly of ribosomes. That would be a much bigger deal, and we haven't followed up on that. But yeah, so I can't say anything really quantitative about ribosomal RNA other than we kind of observed the unprocessed stuff in the neurites.
And the Halo tag tool, you have a distance of reach of the labeling.
Yep.
Is that at all sensitive to environment? I'm particularly thinking of liquid-liquid phase separated vesicles where such a tag might be used.
Yeah, that's a good question. I don't know. I will say that we did successfully do the tagging in the nucleolus, which is, sort of phase separated, I would say. So it could work within a phase separated granule, but as far as the label being able to cross the boundary, I don't know. They're singlet oxygen radicals. So it's, literally, it's O2, right? So it's super small, diffuses very, very rapidly. So I would think it would be able to, but we haven't tested that.
I'll say, to get to the question about why there may be ribosomal assembly intermediates, Melissa Moore and Vivian Budnick had this paper maybe 10 years ago about budding out of the nucleus as a way to get messenger RNAs out. And I remember talking with Melissa about this at the time. Those vesicles were large enough to incorporate spliceosomes or other very large things. So I guess I'm wondering if potentially you're capturing things that have maybe escaped the nucleus through an alternate nuclear export mechanism.
Yeah, that's very much possible. Yeah, we haven't really followed up on that just because it's a little bit too high risk for us, I think, at the moment. But yeah, I think that's something very, very interesting.
Any other question back here? Let's go to Sierra first, then David.
Hi, awesome talk. My question is, are you able to figure out whether or not these regions in the 3' UTR are modified?
You mean like M6A,
Yeah.
stuff like that? Yeah. That is a great question, and it's something that I really wanted to do. There was a paper that came out a week ago, maybe 2 weeks ago, kind of looking at that, and they did see that modified RNAs were more neurite localized, and using some reporters, I think showed that modification was necessary for efficient transport. So I think there is some connection there, and like I said, it's something that we wanted to follow up on. I mean, it's kind of been scooped now. I mean, we're not scooped because we didn't really start on it, but yeah, I think there is a connection there.
Hi, first of all, great talk. So I was wondering, is there a reason you didn't use HaloSeq for the neurites themselves?
Yeah, so two reasons. One is that we already had this other system up and running. It's sort of predated HaloSeq, right? The second reason is, and this was surprising to me, and again, this probably goes back to me not being a neuroscientist. So for HaloSeq, you got to fuse the halo domain to some kind of bait protein that's localized where you want, right? We could not find a protein that was specifically localized to the neurite tips. Now, we could find proteins that were definitely enriched there, for sure, but there was still a lot of protein that wasn't there. And actually, if you think, because the neurite tip is very small, if you think about the total number of protein molecules, most of them were not there. And so actually, when we go back and look at primary neuron IFs and stuff like that, that's, again, the case. So there was no bait protein because ideally, we want every molecule at the place where we're going to be looking at. And we couldn't find a protein that fit that description.
And then also one other question, sorry to go a little bit more into neuroscience. Is there a way you could actually depolarize your neurites so you could see changes between localization when it's...
Yeah, so this is one of those unfortunate experiments that I did as a postdoc, and there was interesting stuff there, but we just never followed up on it. So what we did That was back in the day where we was me. We depolarized these cells just with KCl for like, I think 2 hours or something like that. And then asked what changes in terms of localization. There were a whole bunch of changes, and very interestingly, as a class, RNAs that encoded subunits of the proteasome were way more neurite localized after you did this prolonged depolarization. And if you depolarize them for a very long time, the neurites start to degenerate. So we think, I mean, this is very speculative, but then maybe that's kind of a priming of, I'm going to make more proteasomes here because I'm about to tear this whole thing down.
Thanks.
I guess I have one more question. So if you remove the top motif from the ribosomal protein mRNAs, like if you delete it with CRISPR in the epithelial cells, do the cells care? Like, is there a phenotype if the mRNA is no longer localized?
That is a great question. We don't know that. So the MPRA paper is in revision right now, and we got asked to do that exact same experiment, but with the 3' UTR motifs. And potentially the neurites are shorter if we get rid of some of those target motifs within some of those RNAs. I would expect well, I'm not sure if the phenotype for the top motifs would be stronger. It might be stronger because ribosomal proteins seem really important to me, but it's like all of the ribosomal proteins have that motif, right? So you may, like if you just delete one of them, does it really matter? I don't know.
Well, if it's important for the ribosome,
right. Yeah. So I mean, like I said, I'm on the lookout for any time we can show that any of this RNA localization matters. So phenotypes, even if they're cellular phenotypes, I think it's worth looking at.
Anyone have any other questions?
I guess like with the LARP, right? Rather than deleting the motifs and the protein, and I recognize it might not just be the ribosomal RNAs, but it's presumably acting in RNA localization. Are there mutations that you can make there and ask about whether, more generally, you see a phenotype?
Right. So if we knock it down in the neuronal system, neurites are shorter. When we knocked it out in the epithelial system, there was a clear growth defect. So doubling time was like 2 or 3 times longer. We are interested in rescuing this LARP knockout so LARP1 is a direct target of mTOR, and I didn't get into any of this, but these RNAs are dynamically relocalized in response to nutrient availability, and that requires the top motif. And so we think that happens through mTOR. And so what we want to do is rescue the knockout with, phosphomimetics, phosphomutants to try to put that together.
Anyone else? Great. If not, let's thank Matt for the wonderful seminar.
Thank you.
It's absolutely terrific.
Oh, thank you. Thank you.
Wonderful.
Thank you.
That was really nice, and I can
thank
see
you.
you're not neuroscientist.
Yeah.
But I would love to talk more though.
Yes.
Too bad I didn't really look at your email until yesterday. So